We are almost ready to develop a fast algorithm for finding the two-break distance. The only thing left for us to accomplish it is to prove the two-break distance theorem. Let's consider an arbitrary transformation of genome P into genome Q. We don't know what two breaks accomplish this transformation, but what we know is that during this transformation, the breakpoint graph of P and Q changes into the breakpoint graph of Q with itself, which means that the cycle number of P and Q is changing into the cycle number of Q with itself, which in turn is simply the number of blocks in Q. Therefore, whatever transformation of P and Q accomplishes it, the number of red-blue cycles increases by number of blocks in the genomes P and Q minus number of cycles between P and Q. And therefore, the next question I will ask, how much each two break can contribute to this increase in the number of cycles? Let's look at this. Well, there are different scenarios depending on which two red edges from the breakpoint graph two break affects. Every two break removes two red edges. Consider first the case when these two red edges belong to the same cycle in the breakpoint graph. In this case, shown on this slide, two red edges are removed, substituted by another two red edges, but the cycle number doesn't change. Here's another case where the same two red edges are removed from a single cycle in the breakpoint graph, but the cycle number this time increases by 1. And now let's consider the case when two removed red edges belong to two different cycles. You can see that in this case, doesn't matter which two red edges replace these removed red edges, the cycle number doesn't change. Actually, the cycle number decreases by 1 in this case. And therefore, each two break increases the number of cycles by at most 1. We almost proved this theorem, but actually we haven't proved it yet. My previous slide was simply an invitation for you to look in the picture. Let's give a little bit more formal proof. So a two break adds two new red edges and thus creates at most two new cycles in the breakpoint graph. On the other hand, it removes two red edges and thus destroys at least one old cycle in the breakpoint graph. And therefore, the change in the number of cycles is at most 2 minus 1, which is at most 1. And on the other hand, if you watch carefully my proof, then you notice there is always a two break increasing number of cycles by 1. And here it is, the central case here present an example of such increase. And now the two break distance theorem. We saw that each two break increases the number of cycles by at most 1. On the other hand, there exists a two break increasing the number of cycles by exactly 1. On the other hand, we saw that every sorting by two break must increase the number of cycles by number of blocks between P and Q minus number of cycles between P and Q. And therefore, the two break distance between genomes P and Q is simply number of blocks between P and Q minus number of cycles between P and Q. And armed with the theorem, we actually can compute the two break distance between circularized human and mouse chromosomes. We know that human and mouse genomes can be decomposed into 280 synteny blocks. Each of these blocks will be larger than 500,000 nucleotides in length. The breakpoint graph on this block, if we look at this, actually contains 35 cycles. And therefore, the two break distance between human and mouse equal to 245. Remember this number because it will be important in the next section. Now, there are numerous 245-step scenarios, and we do not claim that we know which one of them is correct. Moreover, the true scenario may have more than 245 steps. Important thing for us is to remember that there were at least 245 steps to proceed to our next section, where we will analyze the random breakage model.